I think we're live. All right. Hi, everyone, and welcome to episode 12 of Behind Our Science. This is our second live episode. We are having Fluorescence Friday live, which is a great initiative to actually talk about fluorescence, and we have a group of real experts in the field. The other thing that we are trying to celebrate is Hispanic Heritage Month. Mm-hmm. We have done it in the past where we highlight people that are either identify with Hispanic community or identify as Latino, Latinx, all of the different communities that we have overall. But what we try to do is try to highlight the initiatives that we have, and we will have some postings for this initiatives, as well as highlight some scientists that we will having in our social medias. Let's start by introducing Vic and Daisy. How are you guys?
Good. How are y'all? How are you?
Not bad.
I guess one thing I want to say about Hispanic Heritage Month is that it's a great opportunity to recognize the contributions from historically marginalized communities, including the Hispanic or Latinx community. So definitely take the time this month to look up like different scientists from this background or cultural backgrounds that have maybe contributed to your favorite topic.
Yes, and check out our last year's episode celebrating Hispanic Heritage Month, which was a live episode as well. And that was super fun. And we got people from all around the world to contribute. So that was really great.
Yeah, that one was really fun, where we got to know a lot of people from different places in the world. We'll try to do it again. Right now, we wanted to highlight some great scientists that we have in the panel. So no further ado, Vic, do you want to introduce them?
Yes, I'd love to. So we get to have some of our really exciting and amazing scientists that we've actually interviewed before, Doctors Mindy and Amy Enjevich, who are both assistant professors at the Medical University of South Carolina. Welcome, y'all. Thank you for joining us again.
Thank you for having us.
So Amy, Mindy, thank you for being in the podcast. I know that you've already been in the podcast to do your solo episode, trying to highlight you guys as some of the very interesting scientists that we have at ASIP and some of the communities that we are a part of. But today we want to be able to show one of the major, I guess I would call it an impact, because you guys do amazing science, and specifically with immunofluorescence. When I see that, I actually feel incredibly jealous. I don't know how you guys do all of this thing, because to my knowledge, I mean, I'm not that good. I always have to ask for collaboration to do IF, and so you guys do amazing work. But today we want to make sure that we are highlighting you guys and the actual questions that we have from people that have reached out to us, as well as we have some live questions that we hopefully get some insights from you guys.
You're too kind, Roberto. Thank you. Yeah, that's very flattering.
I'm not saying that just because, I mean, I really feel jealous when I see it. I don't know how to do that. This is like part of mine. Like my background is actually some that we've done, but I got to say, it's not my work. It's collaborations that we do.
Well, I hope that you'll be able to do some immunofluorescence after this episode.
Oh yeah, yeah, for sure.
Spill all of our knowledge.
Yes, thanks. And so we have, with that in mind, we have Daisy's going to help us with the first set of questions. Again, it's something casual. We just want to get some insights. I believe people will actually benefit a lot, because we have a lot of community that uses ResearchGate, all of these avenues to actually ask questions, so they can probably go back, watch the episode, and actually get some insight from you guys. So Daisy, you want to take it away?
Yeah. Yes, thank you so much, Mindy and Amy, for doing this episode. We regard you as our immunofluorescence queens. So we're really glad for you to be on our episode to answer all these questions. And we've got some questions from Twitter and some people who are dialing in live to ask their questions. At first, I just want to clear one particular thing, and that is something that we just realized. The hashtag, what is it truly? Is it hashtag Fluorescence Friday or hashtag Fluorescent Friday?
It's an excellent question. I use both hashtags. I like using Fluorescent Friday because it makes sense to me, but Amy, I feel like, uses fluorescence exclusively. So I'm welcome to all. I think you could do either one and have it be completely meaningful, but I kind of think that Fluorescence Friday with a C has gotten a little bit more traction. If you use that hashtag, it tends to have more hits because I think more people are using it. But I recommend using both. You cover your bases.
I do use Fluorescence with a CE at the end, but that's just because it's the first one I ever heard of and the first one I ever saw. So I think it makes sense either way, honestly. I don't think it matters, but I go with the Fluorescence just because I don't like change.
And I think the beauty of Twitter and the hashtags is you can make up anything you want. Like Mucus Monday, EM Monday, Microbe Monday, TEM Tuesday, why not White Background Wednesday? I mean, anything.
Yes. Okay. So yeah, basically use both hashtags. I love scrolling through Twitter and seeing all the images pop up on Friday. It just makes my day, my week. Our first question is, what is your best tip for performing immunofluorescence, and what is something you would have liked to have known about it, and that's now fundamental to your work?
Amy, you want to go first?
Okay, so this is probably more than one thing, but bear with me, they're short things. So I think the most important things when you're doing IF is first is your sample prep, right? You need to fix it well and not overfix and not underfix. And you need the right orientation when you're embedding. I also think antigen retrieval is really key. I use heat-mediated epitope retrieval. And if I have problems with my antibodies or my staining, I play around with that. So I'll either use pH 9 or pH 6. If I have a really difficult antibody, I'll use like an anti-fix antigen retrieval. And then I think the other thing that you need to be really pay attention to is your antibodies, the specificity. So that's hard to control because I don't make my own antibodies, at least. So, but the first two ones are ones that's usually what I try to play around with. And if I have problems, those are my first two things I go to. Is maybe I change the fixative of my samples, like instead of formalin or paraffin or PFA, I'll try other fixes that are out there like methanol or glyoxyl. And then antigen retrievals, I'll play with different ones and different timings to see if I can optimize it that way. So those are my best tips, is play with those two steps if you're having issues with your staining.
Yeah. I also have kind of two tips. So my tip is, in addition, when you look at your antibody, when you look online, you see that the antibody looks like it's recognizing what it should, and then you do the stain and make sure that it looks real. But personally, we were working with some histamine receptors, and I saw what I thought was real staining. And then we got the knockout mice, and it turns out that those antibodies were not staining what we thought they were staining, because they were we confirmed that they were knocked out in our mice. So that was really frustrating, realizing that, I'd kind of wasted a lot of time trying to work on these antibodies, and none of them actually recognized what they were. So in the future, I've started reaching out to people that have knockout mice for something. If I don't know that I can trust the staining, I will reach out and say, hey, do you happen to have any just slides that you'd be willing to send me from your knockout animal? I really want to confirm this. I think it'd be really valuable and would help me move forward. And so people are actually pretty friendly. And I feel like we have a good scientific community that really wants to genuinely help each other out. And this is nothing that's going to, scoop anyone. You're just trying to find antibodies, and then they can benefit because they could then have an antibody that is, really accurate and recognizing the epitope. And then during my PhD, I wish I had known about tissue biopsy arrays, where you have all of this different human tissue from different disease states, because I felt like that would have made my work a lot more translatable. And so now that I have, this knowledge, I feel like I can go back when I find certain things in the lab and say, okay, now, is this true in humans in disease settings? And I can easily answer that by going back to a biopsy array and having so many different patient samples. So I feel like that's been really valuable.
Great. Those are really great tips. The biopsy array is a really fascinating one. Is that something that you can reach out to a company for and request what disease you're interested in?
Yeah, so there's actually several companies that do it. The ones that I have recently purchased from is US Biomex. So you could say colon cancer, and they have a bunch of different arrays, and they say, do you want 24 biopsy cores? Do you want 72 biopsy cores? Do you want 180 biopsy cores? And then they're on individual slides. So you can do one slide staining, and you can have, 12 patients, 24 patients, 72 patients on a single slide. I mean, granted, think about the imaging though. You're going to have to image them all. It does take time. So we tend to do a little bit smaller patient numbers. But you can then get a lot of information. They also have like IBD. So they have intestinal inflammation and IBD, and they have, all that's the ones I've been looking into, obviously, but they have a bunch of cancer ones too, like uterine cancer and kidney, clear cell renal carcinoma, and all of these different body sites and all of these different disease states. And so it's wonderful to be able to be like, yes, I would like some normal stomach because I don't know if the staining that I have is real. So I'm going to go stain in the stomach because I know exactly what it should look like there. And it's wonderful to be able to get these human samples. And then I know that what I'm, looking for is relevant.
Mindy, but yet you've gotten good response from like this companies, like meaning you get feedback directly from them saying, yes, no, we'll give you this and that.
No, it's commercially available online. I will put it in the chat. I don't know if you can put it on here, but like you literally just online.
If you send it to us, we'll make sure to put it on the description of the episode.
Yeah.
I think it's a great tool because one of the things that we actually, and I'm going to ask this, we're going off the script, Daisy, but I need to ask. No one's sponsoring us, so I don't care if you call them out or you actually talk bad about them. Which companies you do not like, and which companies do you like for antibodies and overall reactants for IF?
So one of my recent favorites, honestly, since I started my lab is GeneTex. So it's GeneTex. I think it's a Texas-based company. And I've had really good success with some very difficult to find and difficult to stain antibodies. Actually, I think when I first started, I bought about 20 or 30 antibodies, and I think only 1 or 2 I haven't gotten to work, which is an incredible rate of success for antibodies. So I really like that company, and I think they have incredibly great antibodies, especially, we do intestine. MUC2 has been an incredibly difficult antibody to find for mice, and they have an amazing MUC2 antibody. I also really like Novus Biologicals, and Sigma has some really good prestige antibodies that are usually human specific, but often have an overlap in mouse. So those are really good ones. Honestly, I think it's hit or miss on a lot of other companies. You can say Santa Cruz if you want. Santa Cruz Biotech gets a really bad rap, and I agree. Some of their antibodies are just horrible, but they actually do have some phenomenal antibodies as well.
I've gotten a couple. I got to say,
yes,
it's like a 10%. I got to say, like a 10%, but that 10% is like top of the line.
Yes, yeah. They're excellent, and I like Santa Cruz will actually conjugate your antibodies. So usually
they give you samples.
Yes,
they give you samples.
And if you conjugate it, it's a lot. They work really well for the gold mine ones, for the excellent ones. So those are my favorite companies, and I obviously, Abcam, sometimes they have really great antibodies too, but sometimes they have varying success, and it's just hit or miss, which is unfortunate because antibodies are so expensive. But my favorites are GeneTex, and then probably Novus and Sigma.
Yeah, if you give us the information, we'll be sure to put them on the description, because it is important to actually have those done.
I'm taking notes so we can put it in the description.
Thanks, Vic.
I emailed you guys the biopsy arrays already. Second, Amy, I really obviously like very similar companies. I also really like lectins are so easy to use, and they create beautiful staining, and Vector Lab has a whole suite of lectins. So you can just buy these panels, and then you can get them in different colors, different conjugations.
I don't want to monopolize this conversation, but we need to talk to you, because someone suggested a lectin specific for some of the work that we're doing, and I don't know. I've been hearing more and more about lectins, so hopefully what's your experience overall? So lectins, pros and cons.
I really like lectins. I think that they're very easy. You don't really have to worry about the epitope retrieval as much as with antibodies. If they bind hard and strong. I think they create some beautiful staining. I really don't have a lot of negatives about it.
Good to know.
2 thumbs up. Good job, lectins.
Yeah, I want to try out some lectins now, and conjugated antibodies. I've always kind of just steered clear of them, just so like I have that flexibility of colors and things, but it would save like a whole day's worth of work. So yeah.
And they are great if you have, rabbit antibodies are so common. If you have 2 rabbit antibodies, you want to use on the same tissue, you get them conjugated, you can stain them together.
Yes, that's so true.
So it's very helpful, especially because so many antibodies are made in rabbit or mouse. And if you have multiple ones that you really want to see where they are localizing in tissue, you can stain them together.
On choosing different color combinations, I'm wondering like if you guys have a favorite go-to color combo for when you're conjugating those antibodies, and then in post editing, is there a different sort of color combo that you guys like?
I mean, I think we just use the common, Fitzy, Tritzy, far red, obviously the host or dappy color. But I think the second part where you get to be creative and artistic as scientists, I think it's really fun to be able to color, the picture. And I kind of think the picture tells you what the colors it wants to be. You can play around with things, but there's certain ones that like, if the stainings are far apart, like they look better, maybe you can do a little bit similar colors, maybe a pink and an orange where you wouldn't be able to do that if they were right close to each other. And I kind of feel like the staining kind of dictates what colors you use, but I obviously really like the teals, the pinks, the yellows.
So pretty.
He likes a lot of the greens and the purples.
For manuscripts, I always go with cyan for the nuclei and then green and purple for this, just because that's what a lot of microscopists use. And when you have green and purple in certain areas, if they overlap, you'll see white, so you can kind of see the co-localization better. So I tend to go with those colors, but I know, and it's also helpful for people who are colorblind to have green and purple instead of the traditional, deeper blue, green, and red, so that people can see it. So that's what I like to go for. And I usually go for the Alexa floors when I conjugate, or there's the new dye lights as well. So I like 488 and 633, and then I usually use Host and 555 or Psi3.
And how many negative or positive controls do you use? Is there too much controls ever? What's the optimal number of controls for your studies?
It's a complicated question. I think that a control that everyone should do is, the no primary antibody, to make sure your secondary antibodies aren't recognizing anything. So that's obviously a control. I think Amy and I always try to include something we know should stain, like gamma actin or esverin or something that will give a cell shape. And if that looks off, then maybe something's not right with the tissue. Maybe it wasn't fixed properly. That kind of gives you an idea of, maybe it's not the antibody, maybe it's your tissue that's not looking good. Obviously the host and dappy should also stain consistently, evenly every time. And if not, there's something wrong with the tissue and the way maybe it was cut or embedded or processed.
I like to have, so I usually will do a positive control with, especially if I'm trying a new antibody. Sometimes the negative controls are a little bit harder to come by. Like Mindy said, you need a knockout mouse a lot of times for certain antibodies, but I like to do, when I do my staining, I do my control group as well as my experimental group at the exact same time to make sure that everything is the same, that if I forgot an antibody or I used too little or too much of one, both treatment groups have the same amount. So I like to do that. And then I also try to image them at the same time just to make sure they're not fading and not changing over time. But I don't know that you can have too many controls, especially if you're optimizing antibodies. I think it's really important at the beginning to try to make sure that your antibody is identifying what you think it is. And like Mindy said, negative controls are really helpful, but I also like looking at the human cell atlas or the protein atlas. And I also do a lot of Googling when I'm looking for a new antibody or trying out a new stain. I try to see what's out in the literature. And especially if I'm doing, human for the first time with a new antibody, or if I'm doing mouse, I specifically Google mouse intestine staining for this antibody. And I look at all of it, because a lot of times the data sheets that the companies have aren't very good. And it's really helpful to identify if someone has seen this before, that your stain is showing the same kind of thing. But it is hard because some of the data out there isn't great fluorescence sometimes. It's a tricky technique. So hopefully and I think companies are really improving their antibodies, and people are getting better at staining, and microscopes are developing so fast. So I think the field is rapidly developing. And I think in the next 5 years, we'll have tremendous amounts of data out there that will be really helpful.
We have a question from Twitter from @Andy__Osborne. He asks, would love to get some advice on RPE, retinal pigment epithelial cell specific markers for retinal sections, corridor flat mounts. Also, if there are protocols to preserve structure of frozen sections, that would be fab.
So Mindy and I do GI science, so I cannot help on the retinal part, but for frozen sections, I'll tell you what I like to do. Or I think what's really key is that once you fix it, you wash it in PBS, and then I put mine in 30% sucrose. often for 24 hours in the fridge or longer, 48 hours. Once you fix the tissue and you've washed it away from all the whatever your fixative is, then you can leave it in sucrose for quite a while, and it maintains, and that sucrose really helps maintain the structure. But once sucrose is also, obviously, I use 30% sucrose in PBS, and so it's a really sticky solution. So when I am taking my tissue out of the sucrose, what I do is I dab it on either a paper towel or Kim wipe to get as much of that sucrose solution off as possible. And then I take a Weibo and I add OCT to that, and I drag my tissue through it so that that OCT is surrounding the tissue as much as possible, coating it and infiltrating it a little bit, so that it's displacing the sucrose. Because if you put your sucrose solution, or even if you just washed it in PBS from your fixative and put it directly into OCT, you have a lot of water content in there. And that's going to create crystals when you're freezing your tissue, and your OCT isn't going to be fully surrounding it. So you're going to have problems with the cutting, because you're cutting it. When you freeze it, it will be freezing at different temperatures. And then your tissue in the middle won't have frozen just as much as the OCT. So you'll have fracturing. So I like to drag it through the OCT, really coat off as much of the sucrose as possible and get some of that OCT in the tissue. And then I put it in a fresh cassette that's filled with OCT and oriented the way I want. And that really helps, because at least in the GI tract, we have all these villi, which there's thousands and thousands of them. And you really need to coat in between that so that you're not crushing them when you're sectioning them. And the same is true, like with other organs of the stomach, at least in the GI tract, the colon, the liver, it helps for all of my tissue that I try that for. So that would be my suggestion. Some people even do 30% sucrose with OCT in there, so that some of that OCT can get in while you're helping the structure and maintaining the architecture.
Yeah. Wow, that's really great. Thank you. Mindy, you wanna...
I second Amy's. I don't know much about the retina, so maybe Daisy, you have some suggestions. But
yeah, online, we actually got a little bit of attention to that, and people from the field, the retinal field, were actually commenting, and so yeah, we did get some responses, but yeah, I would like the most common that we usually use is RPE65, I would say, and in terms of antibodies specifically, I'd have to go into the manuscripts just to pull them out. But yeah, I would say RPE65 for anti-Osborne. But the tips on frozen sections, that's like so fantastic, because I had no idea about the sucrose. So
yeah, actually, I learned it from a fellow postdoc when I was at Vanderbilt, and I was like, this is genius. And it really improved my frozen sectioning. So I still use it all the time, and it makes it so much better.
It almost looks
so hard. Yeah, sometimes they almost look just as good as paraffin embedded, which is awesome.
Wow. Okay. So now I get to ask a fun question. So I'm wondering what your proudest immunofluorescence image is, and if you could describe it for us.
You go first, Mindy.
Oh man, I'm trying to think. I really like staining bacteria, and I really love these confetti kind of looking bacteria that have a bunch of different stains on them, and they're just really brightly colored, and they to me look like kind of like fireworks. So they just make me happy when I look at them. Sometimes I'm having a bad day or something makes me mad, I just go back and look at my fluorescence and I'm like, oh, that's so pretty. That makes me happy. I feel like an artist. I'm doing more than just failing at science. I'm also being artistic.
So my favorite, well, this is probably, yeah, I guess so. So when I was a postdoc, actually, when I did my PhD, I really struggled with immunofluorescence. I had a hard time. The stomach's pretty autofluorescent anyway, so maybe it was a little harder tissue. But when I did my postdoc, I really did a lot of immunofluorescence. And one of my first images that I took that I really liked was a single villi, and it had like a shorter villi next to it. And I submitted it to the FACID BioArt competition, and I actually got like placed in it. So that was probably like my first big successful immunofluorescence that I thought was aesthetically pleasing. I figured out Photoshop how to like change the colors from the normal, blue, green, red. And it just felt like, oh, I can do this. I think that's one of the things about all of science is that it's a lot of practice and a lot of failures that you have to learn to deal with and learn from. And, you know, if I could show you my first microscopy image, it was blurry, it was horrible. My PI was like, we're not putting that on a poster. So, you know, just give yourself room to grow and kind of be okay with learning the process and getting better every day. And I think that's the best advice I've ever gotten.
Also, sometimes your stains don't work, but they still look kind of cool. I mean, it's not necessarily what you would want to have for a publication, and you're not going to draw any scientific findings from it. But I take pictures sometimes, even if something went wrong and it doesn't look right. I'm like, but that's kind of fun. I like the way that villi kind of crossed over and smeared over here. And, so I take a lot of fun pictures that are never going to go anywhere, but I still think they look kind of cool. And so, that's kind of a fun way to turn lemons into lemonade. It's like, well, it didn't work. And I'm not using that, but at least I have something cool kind of coming out of it.
Yeah, I agree. It's such a learning process, and you can get some pretty things along the way. I think once, like, I was with my friend and we were imaging these tissue sections that she'd just done, and we checked the negative control to start with, and unfortunately, like, it lit up and we were like, oh well, it looks pretty. But yeah. Yeah, so we'll move on to the next portion of our episode. So I'll hand it over to Vic, where she will introduce our live question askers.
Yeah, so exciting. So now we get to have some real one-on-one advice and tips for Fluorescent Friday. So first, I'd like to introduce Freddie Velez. Go ahead and turn on your camera so we can see you and you can ask your question. Hey, Freddie. I don't know if he stepped away. Okay, we can move on. We can do Ward first.
Sorry.
Oh, no. Okay, Freddie's here.
Sorry. Thank you. The question I don't know here was, what's the most number of antibodies you have measured for one specimen? The most number of antibodies you have measured for one specimen. Thank you.
That's right. By the way, gift cards. We're giving away gift cards, by the way. So they're getting one.
Nice. I have done 4 antibodies and a nuclear stain, so 5 stains total. The problem isn't always the antibodies, it's the microscopes. A lot of them don't have filters for the size 7 channel, and sometimes the size 7 is, I would only put my best antibodies that I know are really good in that section because it's harder to detect sometimes. So 5 is the most I've ever done.
4 is the most I've ever done. So Amy's definitely beat me.
Is there something specific about, I mean, I understand there's limitations, but is there something specific that you're looking for when you're combining all of these targets?
So I've done it for stomach, where I was looking for all the different lineages or the major lineages. So I wanted to have the parietal cells, the surface mucus pit cells, the neck cells, and the chief cells all in one panel, just so that I could make, it was mouse tissue and it's easier than doing serial sections to stain them all together. So that's why I was looking at it in that way. And and then another one, I was doing a GFP to replace one of those to make sure that they co-localize with the right lineages. So
how many hard drives do you guys actually own?
Right?
I'm a hoarder of my images. I don't know that I ever delete them. So I have a lot of hard drives full of images. The problem is finding the image I'm looking for sometimes is that, I don't like, which hard drive is that on? So
that's why
like Western Digital. Yeah.
You can post Fluorescence Friday, Monday, like microscopy Monday, just for years. You can just go and be like, oh, look at this one. Let's put this one. Oh, look at this one.
Well, a lot of the images are really bad, for negative data or for certain things, I take the images still, and some of them are absolutely horrible. So they won't see the light of day, but some of them are really pretty. So those are the ones you only ever see the best ones on Twitter. So I've probably for every great image, I have 10 or 15 or even 20 bad ones. So don't think that's not always.
Let me just follow up quickly on this one. So let's say you guys have this, and then now with the new rules for the NIH and reproducibility, you have to have like an actual free repository where you keep all your data and whatnot. how big are you using these repositories? Like they occupy a lot of space. Like Mendeley has options, all of these. What do you guys do?
So it's hard. Our institute uses Box, so we put them in, but at Vanderbilt, I use Dropbox. But as soon as I left, I no longer have access to any of those files and any of those uploads. So I always keep a hard drive, and I also back up my computer and my hard drives to the cloud. So the problem is some microscopes, I used one at Vanderbilt, which I think was the lattice light sheet, and the file sizes were gigabytes and larger. So I would have from a single experiment 100 to 200 gigabytes for just a single imaging session. So, I think that's one of the things that as microscopes advance and they have these huge pieces of data that are being generated, is where to store them. Institutes, I know Vanderbilt was doing a lot of things with Dropbox to get enough data to house all of this data that you would, or get enough bandwidth and enough space to house all this data. But I think it's going to be a big issue because, I also use a Leica SB8, and some of the files, if you use the Lightning ones, they're really big. So it is hard. I try to only use it when I really need it, because most of the time the quality that you get is pretty comparable to what you would if you use the high processing or the deconvolution things. So
yeah, we used to have a lot of hard drives. But,
All right. Well, I guess that's a good investment, to have some hard drives. So I actually have a follow-up question on this naming, finding figures. Like, I imagine it's like when I try to search for a specific email in Outlook, where it's just like, it's like, not today, you could look another day. So do you have any tips or advice for naming your images, or conventional tips just in general for files so that you can find the right one, especially during crunch time, during grant resubmissions or something?
So I always label mine with the date, the tissue type, the objective that I have, and the stains that I have. And Amy actually has every single one of her mice individually numbered, so she can follow every single mouse. And now I'm going to start doing that because I think it's brilliant, but I most definitely did not in the past. But so, I'd have like, this wild type mouse, number 1, 20X, Muck 2, Muck 1, Ezrin, and then of course you don't just take one image of that mouse tissue, then I'd have number 1, 2, 3, 4, as many of the spots that I took on that single slide, so that every slide could get grouped together, and they always have the staining that you did and the objective that you took it at.
Yeah, so I label my mice really well when I do my tissue collection. So I have a lot of different strains. So I have each strain has its own code basically. So I study myosin 5B. So I have a germline knockout. So I label that and with the person's first initials, so like for me, I would have my AE-M5B-1 through all my numbers. And then I keep a separate log, like which ones are knockouts, which are controls. And then I had, a liver specific one. So I say ALB for albumin Cree, and then M5B. And so I have identifiers. So if I look at a block or at this slide, I can immediately tell, oh, this is a liver specific knockout. This is intestinal specific. This is germline. And I think it's hard to come up with the system, but it's a lifesaver because I can go back to my postdoc without even having all the files and just look at them and tell what mouse they're from, what type of strain at least. And then I label when I do my images, I follow that same labeling. So if I know what I'm looking for, like a germline knockout mouse, that's, so I Google M5B, or, I don't Google, I put it into my hard drive and I say M5B, and then I also label my stains like Mindy says, and I usually tell what color they're in. So if I have similar staining areas, like if I do brush border, so it's Ezrin and actin, I say actin green, Ezrin this. And I don't put my objective because I always save the microscope files, and that saves all the data like, time, exposure rates, objectives used, and all of those things. So I usually just label the mouse, the stains that I used, and I also put the date like Mindy.
Okay, that's good. I'm glad you mentioned that you started that in your postdoc, because I just became a postdoc and now I can start that. And in the future, think myself.
Yes. So I got this idea from another postdoc too. So like all these ideas are not mine. I just benefit from having great colleagues and people who really, you take from every person what you learn, and it helps your science get better, and organization is incredibly helpful.
Yeah.
especially when you're starting out. So everyone start out on the right foot and your life will be a lot easier than, having thousands of blocks and trying to find one that you're like, I don't know, maybe it's number 5 or number 10 or 15 or, or maybe that 6 is actually an 8.
Yeah. Were you going to say something, Mindy?
Yeah, I second those things. I was just saying that the objective's nice though, because then you don't have to go back to the original file and then in the file, put in the scale bar. If you know the objective, you can do that yourself and not have to go into the program. But I mean, either one, Amy's completely right. If you do save the microscope file, it has all of the information in it. So you don't necessarily have to.
Usually you can tell too, though, if you do enough. I can tell exactly what 10x, 5x, 20x, 63x.
I'm not there yet. Maybe one day. All right. So our next one-on-one question and gift card winner is going to be Ward Liu. If you don't mind showing us your gorgeous face and asking your question.
Hi, can you see me?
Yes.
Awesome. So I guess my question gonna be how important it is to include like antibody information, publication, and how much do you typically include, like how deep, how thorough do you actually, go into?
A lot of journals are requiring that you have the antibody, like the catalog number, the source, and the dilution. And I think that is incredibly valuable. There's nothing worse than going through a paper and being like, they use an antibody from Novus. And you're like, but which one? Novus sells 4 of these. And I have no idea what concentration you used. And now I have to go figure it out. So I think to help the community, all of us scientists should be as detailed as possible in our methods to make sure that it's reproducible.
I agree. I always hate it when I find a good antibody stain in a paper and there is no reference, or like Mindy says, it says Novus, and there's 8 antibodies that they sell that are rabbit anti-actin, and it's just so frustrating. So I always put the concentration, the catalog number, like Mindy said, the company, and I also keep a log of all of our antibodies that have the lot number on it. So when I aliquot them, it has the lot number because there is variability. They use different animals depending on if it's polyclonal or monoclonal, or so, and there has been a lot of data, I think, in the past that's shown that sometimes you can get a really good antibody from a specific rabbit, and when you make it again, it's not as potent or it doesn't work. So lot number is really, really helpful, especially if you recently changed the lot number and your antibody stops working. If you can call the company and tell them this, they're going to be more likely to listen to you than if you're just like, I don't know, maybe it's the same lot number, maybe it's not. So I always record in our online database what the lot number is for all the antibodies that we have.
And whether or not that antibody worked, so that you don't go back to the antibody that didn't work and try it again,
And I also try to include the antigen retrieval that was used, because sometimes it's pH 9 versus pH 6 is the most common, citrate buffer. So I try to include that in manuscripts because it's really helpful to know.
I've never included that, and yeah, I think I might have to, because I did have to switch between the citric acid, like pH 7, and then the tris EDTA, that's pH like 9.5 or something. That's okay. Good note. All right, so our last question or one-on-one advice tip is from Nathaniel Lardy. So if you don't mind showing us your beautiful face.
You're muted.
Can you hear me?
Yes.
All right, my question is, so in the project, you want to monitor translation of a particular protein across developmental stages in the mouse, various embryonic stages, and also in adult life of the mouse. So you knock in a GFP, green fluorescent protein, such that anytime you translate this protein, you're going to make GFP. And so you are able to track translation of this protein. This is because this protein is very stable and it may mislead you. So you want to monitor its active translation. But then you want to stain this protein and you have very low signal. You want to stain GFP, which tells you that the protein has been made actively, but then you have very low signal. So what would you then propose or advise for such a situation?
So when you paraffin embed, you lose your fluorescence, but if you do OTC, you can, when you cut, everything will still be the fluorescence. So the fluorescence you see if you're cutting it open and doing like, intravital imaging, you would see the same thing in a frozen section. So if you're, rather than having to do an actual stain to like with an antibody that recognizes GFP and you're not getting a good signal, I would just put it into OTC and cut it and then look. You just literally add DAPI just so you can have a reference, and then you should be able to see staining or, fluorescence, not staining, fluorescence if your protein is there.
You can also, there's a really good GFP antibody that is goat, so you can stain it with other things that I believe is Novus. I can find the catalog number and put it in the chat. It's a fantastic antibody. It works really good in paraffin embedded sections. I used it for a GFP knock-in in chief cells. Like when I was talking about the 5 stains, that was one of them. And it was highly specific. It's been reported in a lot of different studies, and it's a great antibody. And I think it's like a 1 to 2,000 dilution or something. So it's really potent as well. So I'll look that up and I can send it to Vic, or hopefully if I can get it soon enough, I can put it in the chat.
Right.
Yeah, no, I can definitely send it to me.
Can I add a follow-up question?
Oh
Yeah,
yeah.
All right.
No more gift cards though.
That's fine. All right. So yes, you're right. OTC would be preferable. We have FFP samples that we retrieve the antigens. One other method we have tried is to look for antibodies against GFP, but then that gives us very high background.
So is the question getting rid of the background? Amy, go ahead.
So you can do the GFP antibody that I just mentioned. It worked really well in IHC, highly specific, really low backgrounds. You have to be careful with IHC about your developing time, but I think when I did it, I think it was 15 seconds. So it was incredibly rapid. So you have to add it each slide, obviously wash it off as soon as it's developed, and then keep all of them the same amount of time. So I would do, if you have a GFP that from, like Mindy said, you can get a positive control from someone else who's published on a GFP knock-in animal or knock-in cell line, and make sure that your antibody specific and you're not oversaturating it. I would suggest trying that. But the antibody that I've used it in multiple knock-in mice for GFP, and it's worked incredibly well, and it has a really low background actually. So I would recommend trying that. Or like Mindy said, OCT would also work well, but sometimes I think OCT is a little bit trickier to do, as far as I personally prefer paraffin, but I think your architecture is better. And if you use a good antigen retrieval, I think you get a much better stain as well. But that's kind of against the dogma.
Yeah, cryotomes are tough. I feel like they're tougher to cut with than microtomes. And maybe it is the temperature. I don't know. Thank you so much, Nathaniel. All right, so I have one more question. We're going to kind of rewind and go back to basics in case we have any followers or listeners that are trying to figure out if they want to do an immunofluorescence or immunohistochemistry. Could y'all comment on the differences between staining and labeling? Because it's so important to have the right words whenever you're trying to do an experiment, especially when you're looking at protocols online. So
like when usually for me, when I'm labeling something exogenously that I'm going to be adding to like a cell. So like, for instance, I'll fluorescently tagged bacteria that I then add, and then they bind to cells and I fix them, and then I will, cut them and then do staining for something else. And I'm using an antibody for something that, I'm adding onto a slide. So to me, that's what I view labeling being outside, adding something fluorescent that I'm going to then add to something else. Maybe Amy's more articulate. I'll bet you are.
I think when I think about it, I would say staining is something that involves an antibody. And I think labeling is something that's either like a host or a DAPI or even a lectin, right? Because it's not technically an immunoreagent, and it's already usually pre-conjugated. And so it's already labeled as well. And so I think of stain like immunohistochemistry or immunofluorescence as antibody derived, and then labeling as non-antibody, but there's similar pathways that you're using basically, right? You're still conjugating a lectin, or you're staining the nuclei. or, you're labeling, usually I use lectins. So those are the only ones I can think of right now.
For like IHC, you could use like, Alcyon blue to
Yeah, I would think I would consider like IHC. Well, there's so many different kinds. So maybe, I don't know. I've never thought about it actually. That's a tricky question. And I honestly think probably it just depends on, the scientists you're around and the vernacular that they're using. It's probably more jargon than anything. And I think people probably use it interchangeably. And so I don't know that there would be necessarily a right or a wrong, but I kind of agree with Mindy that I think of something labeling as exogenous and something as, immuno-derived as antibody derived.
Got it. That's a really good distinction. Thank you guys so much. We actually have another quick question from Nathaniel. He wrote it in the chat. He was wondering if y'all could comment on signal amplification during your imaging process.
So signal amplification,
you mean like,
I wonder if he means intensity, like when you mess with the intensity or turning your blacker kind of thing.
Want to jump off again, Nathaniel, and ask the question?
yeah, I'm assuming that you're saying like on the
When he
microscope,
comes back, you can explain.
increase the laser to get up, to get it to be brighter.
I think I overdo mine personally. I tend to go on the high end. I like bright stains personally. So I also think I oversaturate my antibodies. I tend to use, if I'm using a new antibody, I tend to go 1 to 100 or 1 to 50. So I go on the higher end so that I have maximum signals. And when I use my secondaries, I also go to the higher end of the dilution, like the more concentrated ones instead of dilute. So I think I like to get as bright a picture as I can, because you can always dim it, but brightening it often adds background.
I also think too that it's important if you're doing like wild type versus a knockout mouse or a treatment group that you first scan your images by eye and look for the one that's the lowest, and then or the high, like either one if you're going to go too high or too low, and try to make it so that you find the right setting where you can get good looking images for your different conditions, so that one of them's not just blown out and so high and the other one's not so low that you can't see anything. So I think it's important to make sure that, before you start imaging, that you just by eye look and make sure that, it's not like this one's flaming hot and this one's really low and you amped this one up so much that now that by the time you got to that one, it's going to be crazy staining for the other treatment group.
Nathaniel, does that answer the question, or do you think it might be a little different?
Or are you thinking like afterwards you have an image and you're like, this isn't bright enough, I want to take Photoshop and go like this and make it brighter?
No, I mean you have very low fluorescence, so you want to boost the signal using any of these commercially available kits like Theramite, Super Boost and amplification kit. I mean, that is what I.
Oh, I've never used a kit. I've thought about it, but I haven't tried it. That's a good idea. And I don't know how successful they are, but I imagine, a lot of the kits that companies are selling, they're usually pretty good and they sell them for a reason. So people must be using them, but I don't have any experience personally using them.
Me neither.
But I might have to order some and try that. That's a really good idea.
For the next time you guys do a Fluorescence Friday live.
All right, perfect.
We'll try it
next year. All right, thank you so much, Nathaniel. So we have a couple minutes and I would like to do some rapid fire questions. So I'm gonna name a question or give you two choices and y'all need to let me know which one is your choice. All right, so the first one is monoclonal or polyclonal antibodies?
Monoclonal.
This is probably not accurate, but I like polyclonal.
Amy, we're gonna
polyclonal.
All right.
Better signal.
IHC or IF?
Oh, IF.
IF.
PFA or methanol?
PFA.
Methanol.
Cryo or paraffin? So OCT or
paraffin. 100%.
Mindy?
Oh, I said paraffin. Sorry.
Oh, okay. Sorry, I didn't hear you. My bad.
Paraffin.
594 or 488?
488.
Oh, 594.
All right. Tritzy or Fitzy?
See.
Tritzy.
All right. Dappy or Hosh?
Host.
Host. Okay, I mispronounced it too. Great. So now we
Oh,
understand.
I don't know how to say it.
Hold on, Vic. Is it bad that I actually disagree with most of theirs?
Wow. Wait, but didn't you
Fair.
say that you don't do your own states?
For some, I do. Don't say that out loud. Pitt is hearing us. No, I do. But I mean, there's some specific. Like I always do OCT. Maybe it's because of the equipment that I had. But yeah, it's also a limitation. So it's good to know. At least we know that there's like even different opinions on like people that really know what they're doing.
And
Yeah.
I've been saying host wrong. How do you say it again?
We might be saying it wrong. So
I think I say it wrong. I don't know. It's
During my PhD, we were saying Herxt.
That's
It's like completely
that's bad.
different. That's
That's
why
not-
I was like, that was like one of the things I wanted to clear up as well.
as host. I don't know.
yeah, I call it
That
host.
makes sense.
Yeah, you also say ASIP, Daisy. So
Oh, it is
I
ASIP,
do.
though.
It is ASIP though.
yeah,
ASIP.
Proportional to your age of how you pronounce it. There's a linear relationship.
Oh, I see.
correlation.
Wow.
R squared equals 1.
No, I think that was great. I think we learned so much. I actually say I would have to go. I'm gonna go watch the episode back and then actually take some notes because it's really important that we have at least an insight, because many of us sometimes, like, you're giving instructions, you're starting with all of this, and sometimes it's hard to actually do so, and sometimes we don't have as much of a network to do so. So it'd be a good idea when we have all this insights from people that actually know what they're doing.
Well, shout out to Amy. She wrote a book chapter and gave all of her protocols on how to do the best staining of all time. And so hopefully that will come out in press
Yeah,
sometime.
put it on the description. We'll put it on the
Oh
description.
yeah. If you let us
Yeah,
know,
we
we
can
can.
share
We'll
that.
start like advertising for that. That's awesome. Congratulations.
Yeah,
Oh,
congrats.
thank you. It's very detailed. So like probably
Love it. Yes.
We love the
Snap. That's awesome. Well, thank you both so much for your time for another Friday night with Behind Our Science podcast. Like, thank you so much. I can't remember if the last one was on a Friday. I just remember you guys dedicated your time. So definitely double the thank you. If y'all want to follow them, we'll have their Twitter handles in the description of our video when it's posted on YouTube. They are very good about posting their Fluorescent Friday or Fluorescence Friday images. So definitely a nice treat. I have myself saved a few for backgrounds for things. So I hope that that's okay.
perfectly
All right.
flattering.
So I also want to advertise that next Friday is the PISA 2022 Young Investigator Virtual Conference. And I was very lucky and I got to chair the planning committee, and we hopefully have a really good lineup. There's also some career development sessions that will be coming out. There's going to be a virtual hangout with other trainees, including Daisy and Roberto, and Roberto's the special guest star for how to
Yeah.
find the right PI in lab.
It's a surprise. They don't know.
Really important question. Like, some of us go through unfortunate situations, or maybe you're in a really good situation and you can share your advice and tips on how you found that perfect lab. There's also how to obtain funding, because we all are chasing the money bag and we really need it to do the studies we want. And the last career development session is the PhD to postdoc transition, because as everyone on this call knows, it's a really weird ethery space that you're in. You're a trainee, but you're also not. And it's good to get advice on how to navigate these situations. Be sure to check out at ACIP Path to find out more, and you can register online through the link on their Twitter page. And with that, we can conclude it. It's exactly 8:00. We did it.
Oh, this is amazing. How do we
Yeah,
do-
thank you guys for being in the
Absolutely.
podcast. We also have to say this is our 12th episode. We actually didn't set up a landmark when we hit the 10 episodes. So we have to give a shout out for our editor, Jeff. I actually so Jeff is our editor. Has been great coming in and then putting us into a very different perspective and having us chase a dream about doing the podcast We also want to thank the people that actually sponsor, support, and help. Spotify, we're almost in every single one of those audio platforms.
Mhm.
Apple Podcasts, Spotify, iHeart, all of those. And we appreciate the people that collaborate with us, the People of Pathology podcast, and all the different venues that actually appreciate and actually value what we're doing. So we appreciate the opportunity to do this for you guys. We'll be able to keep that in mind. Happy Heritage, Hispanic Heritage Month to everyone that's celebrating and whoever identifies as Hispanic, Latin, Latinx, and everything that comes together with it. So thank you guys for all, and we'll see you on episode 13.